We want to thank the organizer for putting together this great meeting. I've learned a lot, and also thank you everybody for sticking all the way to the end. Hopefully it's worth your while. So I'm from PowerBio, which is a small company located in the Bay Area. And what we're really focused on as a CDMO, CRO, is to really help amazing companies, such as some of them here, to develop their bioprocess, as well as to do initial pilot scale, demo scale, scale up. And what we're really focused on is to develop continuous biomanufacturing as a platform. Next slide, please. So this is something out of McKinsey a few years ago now. Biomanufacturing is poised to disrupt a whole range of industries, and the Biden administration is very supportive of this effort. But in order to realize this circular bioeconomy, there are two major problems we need to solve. One of which is that if we to the capacity to make this gigatons of products, where are we going to find the fermenters for those? And the other is to produce it at a cost that is competitive with these industries that have centuries, if not longer, to really optimize their processes and really drive down the costs. And as Joe from the Avery Company alluded to, what consumer really wants is first is quality, the taste and the texture. But the second thing that they care about is really the cost. So in order for bioprocess, our bioalternatives to be competitive, we really need to improve the cost of those productions, particularly in the commodity markets such as chemicals or food or agriculture. Next slide, please. So this is a typical journey, to take a bioprocess from a great idea all the way to commercial scale. And a lot of focus and innovation has happened in this within the last decade or two in this space with string engineering and design, technologies such as CRISPR and all of the really amazing cost savings from DNA synthesis and so on. But very little has been done on the process development and the production side. And really, we're using technology that are a decade, if not a century old, and that's really fat batch and fat batch fermentation. Next slide, please. So Laura from Sunflower Therapeutics has done a great job of explaining why continuous fermentation is such a great process, with the quality of the product as well as the increase in productivity. So I won't belay the point, but I say that the traditional fat batch is very slow, repetitive, whereas if you can produce continuously, gets much higher productivity and so on and so forth. And the chemical industry, for example, have really done away with batch processing, and pretty much all of the commercial chemical processes are all in flow reactors because they're just so much more efficient. And if bioprocess is ever to compete with that, we really need to develop a new manufacturing platform and really go towards the continuous route. Next slide, please. So continuous fermentation is not a new idea. It's been around for a while. It's been called the gold standard of biomanufacturing, but it hasn't been realized in the commercial scale except for a few cases. For example, ethanol production in cerevisiae or lactic acid, really products that the microbe naturally makes. In my previous life, before joining Power, where I was a string engineer, we tried to engineer ethanol production out of cerevisiae, and it was such a nightmare. They just do not want to get rid of that. But in the processes that we're talking about here, whether it's making egg white protein or antibodies, we're really asking the host microbe to produce something that they don't naturally want to make. And that really put a very strong selective pressure on them. And most of the continuous fermentation that have been tried in the past have really occurred in one single reactor, so something like a chemostat, where you're asking some of the cells within that tank to grow, to replenish your biomass, while at the same time also making products. And this really leads to what we call the cheaters, cells that have lost their production capabilities to really just outcompete the culture and really, you lose all your productivity very rapidly. And we even see that in just even fat batch culture, where you have a beautiful green GFP culture, and then come back the next day, and all of those cells have lost their capabilities, and you just find a muddy black culture. So how do we stabilize the strain to make all of these amazing products in synthetic biology and precision fermentation? And what occurs to us is that cells that are actively dividing versus when they're making a product, they're really in very different metabolic states, and the conditions that are optimal for each one of those is actually very different from each other. So why are we asking them to do that all at once in a single tank? And so, while it seems like a simple idea on the surface, what we have done is just physically separated those two really different metabolic states. So we have a tank, what we call the co-change growth stage, where the microbe, all they're doing there is growing. They're strongly repressed from making any product. So there's very little selective pressure for them to lose the production pathway. And those cells are, those happily growing cells are continuously being transferred into a 10 to 50 times larger production stage. And here we run this production stage close to a new term that I learned from this meeting, a retentostat, where we provide them with sufficient nutrient for them to remain metabolically active, have all of the stuff to make your particular product, but they do not have enough nutrient for them to grow. And we're aiming for, for the doubling time here to be in the order of days to week, but not a true retentostat, as we're continuously supplying fresh cells into the production cell so that we refresh that, so that we can get the increase in productivity. But we do have a cell bleed so that to remove the cells, otherwise, our culture really turns into mud. They have, ODs that are up into the 600-700 range. And then we have a, similar to what Laura described, a cell retention device on the other end to really recycle the cells back so that we can intensify the process and get the higher productivity. For us, we put this outside of the fermenter, but we configure it in such a way that really the cells are spending less than a minute outside of the cells, and when they're in this production stage, where they're more quiescent, they're actually pretty tolerant to these stressors, and we haven't really found any effect of having them outside of the cells. So by putting this together, this 3-unit operation together, we have established a system for precision products where we are much more solve the problem of genetic drift and much more resistant contamination. And we have examined clones out of both of the fermenters after 300-400 hours, and we still have greater than 90%, 95% of the cells have retained their production capabilities. And that's actually even better than the corresponding fat batch process, where we typically see 20 to 30% loss even after just a 100-hour run. Next slide, please. So we just initially started working with Pichia really within the last year, and that's why we're here to really learn from this great community. But in some of the early experiments at the bench scale, here we have taken a Pichia strain that is pretty productive. In the fat batch, they're getting about 2 to 3 grams per liter. But when we convert that process to continuous, we're seeing a 2 to 3-fold gain, and we're running these processes for a couple of 100 hours, so you can run that fat batch process 2 or 3 times, and even then we're still seeing a greater than 2-fold gain in productivity. And this process, I have to say, has not really been optimized. We just converted it from fat batch into continuous, so we think that there's actually a lot more gain that we can have. Next slide, please. So, we just started working with Pichia. We're actively scaling that process now. My colleague who's here with me, Evan, is really spearheading that effort. But we initially developed this 2-chamber process for a related product, which is a secreted molecule in E. coli. And here we have taken that process and we scale it up to our largest fermenter that we currently have, which is 300 liter. You can see the scale-up data there, and the fat batch process for this was I led that along my team in my previous company. We have commercialized this process, and the scale-up didn't look nearly as good as there. In the fat batch process, we were able to achieve like 80% of what we can get at 2 liter scale, but here it's essentially 100%. And we've been able to transfer this process outside of our lab to a national lab, and they were able to replicate the results as well. So we're building this technology really to be bolt on to existing fermentation infrastructure so that we can get the increase in productivity without having to spend the capital to build new facilities. Next slide, please. So the other thing, as I mentioned, the processes I've shown has not really been fully optimized, and as Laura from Sunflower Therapeutics mentioned, one of the great things that we can do with continuous fermentation is that it's really to enable very rapid process optimization. In the traditional DOE, you get 1 data point per experiment that takes days to a week. But in a continuous fermentation, once we get the system under steady state, we can perturb that steady state, read the output, and so we can get a very data-dense experiment very rapidly. And because the experiments that run in steady state is actually very much amenable to automation, so we've taken that idea, next slide please, and build a AI algorithm that would do the process optimization for us. As James alluded to, while AI is very smart in doing a lot of things, they also can be quite stupid. So what we have done in this is to put guide rails around the experiment. So when we initially started working with this, we didn't do that. And one of the things they tried to do was to grow Pichia at 50 degrees C and cook the tank. So here, in our initial phase, we were doing it in silico, trying to get with a simulated response surface, and we're asking the optimizer, which we have named Sophie, to identify the next slide, please. Well, there was supposed to be a little graph of how it explored our surface and reached the optima. But we have taken that now out of the in silico and actually deployed it in tanks. This is a early experiment that we did, again, with a Pichia secreted protein, asking it to look for temperature and pH optima for producing this product. And when it started at some temperature and pH, and then based on the titer feedback in this case, it was exploring 2 different parameters, and very rapidly, within a week and a half to 2 weeks, I was able to identify temperature and pH condition that improved this titer by greater than 2x. And we're now, with experiments that are ongoing in the lab at the moment, really pushing the system, asking it to optimize multi-parameter ones, 4, 5, and as well as looking at a range of different outputs, whether that's titer, productivity, or yield. Next slide, please. So combining these 2 technologies together, what we think we've been able to do is that really reduce the cost of production, and that's really the major driver. This is a TA analysis that was done by a third-party consultant who used to work for NREL to really assess the economic impact of bioprocesses. And here you can see that the very typical cost curve for a fat batch process, and you really have to reach very, very large scale in the hundreds of thousands of liters before the cost curve really flattens out. But for the corresponding continuous power bioprocess, we're able to achieve this flattening really at pretty small scale, and actually even in the 1,000 to 5,000 liter scale, we're already seeing that the cost of production is really much lower. Next slide, please. So really, what we can do is reduce the cost of production from 30 to 50%, in some cases up to 80%. We really can accelerate the go-to-market because we can do the process development so much faster. Because we're using smaller fermenters to achieve the same production scale as you would for a larger fermenter, it makes it much more very nimble and scalable. And as a lot of talks here today in this meeting is about antibody production in Pichia, and we think that with this system, it's much more amenable to be able to be deploy in a distributed system, so you don't need the giant fermenter to produce the antibody that you need all over the world. You can produce it much more locally. And next slide, please. So with that, I'd like to thank the team and our investors. And please come talk to us. We'd love to explore what Power technology can do for your processes, whether that's making an egg white protein or antibody or all of the other amazing bioproducts. And thanks again for your attention and staying all the way to the end.
Questions?
Thank you, Brian. It was a great presentation. If you look at the strains, what do you need to know before you go to your guys to run your process?
Yeah, so we're trying to design a process to be as strain agnostic as possible because make it easier to adapt. Obviously, because we're running these processes for hundreds of hours, strain stability is always a question. But you also need to have the same for you when you're scaling up a fed batch process as well. I think the thing that the way that we design this process is really geared towards precision fermentation, where a process is really the product is really in two different stages, right? It's an induction process where you do something to shift them from just growing to production. If they're growth associated, we haven't really explored that as much, but we think that it works much better for a induction type process where they're already really broken into two separate stages.
So it's possible that you give your clients basically a check sheet and then fill out what the strain can do, and that gives you basically an indication if you can do your process for that particular strain.
Yeah, I think actually what we oftentimes do is what we call proof of concept type of experiments for our client, where they come to us with a strain, whether that has been run in a fed batch or even just a flask. And we would evaluate whether it is a fit for our process, examining the strain stability and the production stability. We don't even need to have that checklist because that's one of the first things that we would check. And if that doesn't work out, then the project doesn't move forward.